now is talk about the molecular explanations of Mendel's postulates, at least 3 of the postulates. We'll save dominance of the molecular reasoning for dominance for a later lecture. But let's see. So let's talk about Mendel's postulate on alleles occur in pairs. Alleles occur in pairs. Okay, so one of the great things about Mendel is he was working before scientists. They knew there were cells. They didn't know what was inside cells. You know, they didn't know there were chromosomes. They didn't know about genes. They didn't know about DNA. So he was working essentially when scientists knew cells exist, but didn't really know what was going on inside them. And he was able to predict how traits are being passed from parents to offspring in peas, you know, just by doing genetic experiments. So he explained what must be happening and left the figuring out the details of how it's happening to future generations. So alleles occur in pairs, and why? What is going on? Why this seems to be true, but what does it mean? So we now know that in pea plants, peas are diploid, and if we take, say, a pea plant that is, say, heterozygous for the plant height gene, we have one big D allele and one small d allele. Well, alleles occur in pairs because peas are diploid. So if we look in a pea cell, say in G1 phase, and I'm diagramming the chromosomes like they're condensed, but they wouldn't be condensed, right? They would be decondensed and there'd be chromatin throughout the nucleus. But I'm diagramming the chromosome like it's condensed so we can see the chromosome and see that at this position here, well, that's where the plant height gene is, and this chromosome has the big D allele, the big D version of that gene. Now, peas are diploid. That means that there's another copy of this chromosome in the nucleus. Those are the homologous chromosomes. There's also other chromosomes we don't care about for this example. We just care about the pair of homologous chromosomes. And at this position up here is the plant height gene, but this one has the little d allele. Okay, so this plant has 2 chromosomes, 2 genes, 2 copies of the plant height gene. One of them has the big D allele, and the other one has the little d allele. So Mendel predicted the alleles occur in pairs, and well, peas are diploid. That's why the alleles occur in pairs in pea, in humans, and lots of other organisms. So segregation. So Mendel said that the alleles segregate away from each other so that, you know, gametes inherit one or the other with equal likelihood. Now, he didn't know about meiosis. He knew gametes were formed. They knew about pollen and eggs and stuff, but he didn't know about gametes, but he predicted that when they were being made, those pairs of alleles, the gametes only inherit one of the alleles. And that's segregation. And we now know that during meiosis, and it's a little complicated, right? Because we have the pairing of the homologous chromosomes, and at this stage of meiosis, the chromosomes have replicated, have already replicated, so they're represented by pairs of sister chromatids. So sister chromatids here, and sister chromatids here. But if we're still working with our plant, that is of the big D, little d genotype, well, the big D's can be over here, and the little d's over here. And I didn't diagram the chiasma, right, that holds these together during meiotic metaphase. But just assume it's there, holding these together. So now this is what it looks like during meiotic metaphase. And this is when the alleles segregate away from each other, meiotic metaphase 1. So after meiotic or meiosis 1, we're going to end up with the D alleles over there, and this product of meiosis 1, and the little d's over here. Let's say that right here, this position of the chromosomes. And then this is going to go through meiosis 2. So the gametes have only one D allele, big D allele, or one little d allele. And you can see these gametes are made in equal numbers. So that's segregation. Now, if we were talking about oogenesis, right, especially in humans, then only one of these is going to be formed, right? But it's still random chance whether this is going to be big D or it's going to be little d. Unless we're talking about meiotic drive, and that's something my lab studies. That's a slightly complicated issue involving selfish genes and genes that are able to bias whether or not they make it into the ootid. They bias them their progression to the ootid relative to the polar body, but those are rare things that happen for the most part during oogenesis. Even if you only get one product from meiosis, it's random chance whether that product would be one allele or the other. Now, I hope I didn't confuse you too much with that aside on oogenesis. So, one more thing I would like to talk about is independent assortment. So we've talked about independent assortment and how it, because of it, all of the gametes that were formed in equal numbers when we're looking at two different traits, right? So just briefly, so independent assortment entails or allows for an individual with this genotype to make 4 types of gametes, and all of those will be found in equal numbers. There's no bias towards one or the other, or one or two over the other. So what's going on? Why does independent assortment occur? Why is there no bias towards some gametes over others? And the reason is that if this, let's say this is one tetrad with the plant color gene over here, the big G alleles on this chromosome, the little G alleles over here, we're in meiosis 1. Now the big G's are going to go that way, the little G's are going to go that way. Well, independent assortment occurs, it's most easily to see it, that it occurs when the genes are on different chromosomes. So here's the plant shape gene. We'll put big W's here, little W's here. Now in this case, if we follow this all the way through meiosis, we will get gametes where the big G's are with the big W's and the little G's are with the little W's. And the reason that is the case is because you can see if this is the metaphase plate, the big G's are aligned on the same side as the little W's. But this orientation of this tetrad relative to this one is formed in a random process. It could have easily, half of the time what would happen, it would have formed the other way around. So the little W's would have been, well, I should just draw diagram 1. A little W alleles on the left and the big W's on the right. So half of the time, meiosis is going to produce these products here. The other half of the time, when the little w's are aligned on the same side as the big G, we're going to get something like this. So if we look here, these guys right here, these are these are each found over here. So we've got this one here, okay, accounted for. That one's accounted for. That one's accounted for. And that one's accounted for. Okay, and now we see the molecular reason why, you know, with an understanding of meiosis and how the chromosomes are moving, we can see why these genotypically distinct types of gametes are formed in equal numbers.